guys, this is Mrs. Schramm. We are here in our packet. This is our new genetics packet with our question, how are these two twins so different? We've done our reading on karyotype, and you should have answered these questions. You also should have read and looked at these two karyotypes, as well as this HeLa cell karyotype, and answered these three questions. We already did this. You should have this done. Remember, we're talking about meiosis and mitosis, and what's the difference? In mitosis, when we were doing our planarian, where we cut our planarian in half, mitosis is the process of making two new identical cells. But today, in this unit, we're going to be talking about how we make sperm and egg cells. So meiosis is similar to mitosis in that it still has prophase, metaphase, anaphase, telophase, and cytokinesis, only it does it twice because it needs to divide, and instead of ending up with two sets of every chromosome, it ends up with one set of every chromosome so that you can pass it on only half of the DNA from mother to child. You should have answered these questions to look at the differences between meiosis and mitosis, and you should have completed this reading where I tell you guys a little bit about gene mapping, and then make sure that you understand these vocabulary words. I provided a picture. You were supposed to provide your own definition. Once you've done that, you're ready for today's lesson. Today's lesson, we're going to be talking about meiosis. To prepare you, please pause your video after you have read and answered these questions, which are on page 12. Once you have answered these questions, move on to watching me and listening to me answer the questions on here by filling this in so that you can properly understand meiosis for our quiz on Thursday. Here we go. So, here is an overview of meiosis. First of all, let's remind ourselves of our vocabulary words that we should already know. You should know chromatin is where DNA is very loose inside of the nucleus. It's not wrapped up tight into chromosomes. It's loosely in the nucleus so that it can be accessed to do normal basic cell things like using the DNA to make proteins. If a cell is going to go into mitosis or meiosis, then the chromatin will curl up inside of the nucleus to form a chromosome, such as this. If the cell is going to divide, it will need to first do DNA replication. And when DNA replication is made, you guys already learned about that, where the DNA splits and then matches up, forms two new identical copies, and those two copies stay attached to each other at a common centromere, right here in the middle. Now, I guess I will label that centromere. Now, over here, what are homologous chromosomes? This is a new vocabulary word. You guys should definitely know sister chromatids. You should already know that if this is a gene like A for brown hair, this will be a gene A for brown hair, okay? However, homologous chromosome pairs are when you look at the one that you got from your mother and the one that you got from your father. So let's say you got this one from your dad, and my favorite color is pink, so I'm going to put a pink one from your mom. Okay. Now let's say that dad has brown hair, which I'm going to put as a capital A. This chromosome pair is homologous to this one because they both have the hair gene, but this one might have a blonde hair gene instead of a brown hair gene. Okay, so they carry the same genes, they are the same length, their centromeres are in the same place, but they might have a different version, which is called a different allele of a trait. Okay, so reminder that homologous chromosomes can have three things. 1, 2, 3. They are the same length, have the same genes, and centromere. Centromere is in the same position. Okay? One thing that they do not have in common is that they may have different versions of a gene. Those different versions are called alleles, okay, of a gene. Okay, for example, again, brown hair versus blonde hair, both hair color, but a different version of a gene. Now, let's look at meiosis. We're going to use, we could sit here all day and try to draw every set of chromosomes. Remember, you have a chromosome 1 from your mom and 1 from your dad. Chromosome 2 from your mom, chromosome 2 from your dad. You can see that the biggest one is chromosome 1, and that down here the smallest one is chromosome 22. And then over here we also have pair 23, could be an X and a Y if you're a boy, or an X and an X if you're a girl. Okay? These are called autosomes, all the ones that we all have. And then the sex chromosomes are the one that determine our biological sex, so for girls and boys. So, we're not going to draw all the chromosomes. So I'm just going to choose 2 random chromosomes. I'm going to say, like, I'm going to draw a big chromosome, like chromosome number 1, and then I'm going to draw, like, a smaller one, like chromosome number 7, okay? So we're just going to follow 2 chromosomes through meiosis on this paper. I'm going to choose 2 colors. Please make sure you have 2 colors to use also. I'm going to choose green and pink. And here we go. At the very beginning, every cell has 46 chromosomes. I'm going to write that it has 46 chromosomes, but we're going to be representing it with only 2 pairs. 1 pair you get from your mother. That's going to be chromosome 1 from your mom. Over here is chromosome number 1 from your dad. Notice I tried to make them the same size and put the centromere in the same place. Next, I also have a chromosome number 7, let's say, from my mom, and a chromosome number 7 from my dad. And I'm trying to make those about the same size, same place. Now, you guys know, before we do mitosis, we're going to have to turn these into sister chromatids. So DNA, or chromosomes being copied, is called DNA replication. Okay, so I'm going to copy the chromosomes. Here we go. Chromosome 1 from mom copied, chromosome 7 from mom copied. We would do it with all 23, but we don't want to be here all day drawing. Okay, we'll do the big chromosome from dad and the smaller chromosome from dad. Okay, now here we're ready to start meiosis, and there's 2 cell divisions that happen. So instead of making 2 cells like mitosis, we're going to make 4 cells. But there's something weird that happens here. These 2 chromosomes get really close together. They become so close that they actually swap a tiny portion of their DNA in an event called crossing over. So up here in this area, we're going to draw a little picture of what happens. And first I'm going to show you a little thing that I made about what happens. So let's say we have chromosome number 1 from mom, chromosome number 1 from dad. And you can see here in my little sticky note diagram that these chromosomes, they get really close together, and then what happens is a little chunk of it gets swapped. There's actually an enzyme that helps do this. So for my purposes, I'm doing it with a sticky note to make it work. Okay, and a little tiny piece of the DNA is swapped. Now this is interesting because this adds to the variation possible between siblings. So you're not actually just inheriting dominant gene A, B, C, and D all from mom if you get this piece. Or, and when I say mom, it's actually your mom's mom. So this would be like from your grandma, or this piece would be from your grandpa because it's from your mom's dad. Let's say your mom's mom had black hair, brown eyes, like all these dark traits, and then your grandpa had light hair, light eyes, light skin. You're not just inheriting that. Because once it copies and then they come together, this crossing over event that takes place swaps a tiny portion of the end of the DNA. And this creates a new combination. So now maybe you get somebody with brown hair and blue eyes instead of like blonde hair and blue eyes. Okay? So it allows us to have new combinations. So it's not just brown hair and brown eyes, blonde hair and blue eyes, but now you could have blonde hair with brown eyes or brown hair with blue eyes. Okay, that's what these genes kind of represent, a new combination here that's possible. This process of swapping, in this case, I'm looking at the Q arm of chromosome number 1. We've swapped a tiny chunk. That's crossing over. So we're going to draw that and write it right here. we will write crossing over, okay, takes place during prophase 1, okay? And we'll draw a little picture of it. We will draw a bigger version of this so that we can see it more clearly. Here's our pink DNA. I'm going to draw this, only part of that one, so that I can draw it being swapped with the other part of the green chromosome. So I'll draw the green chromosome. And then the lower half will be the pink part, okay? And like I drew some genes on there, I'll just say like this is gene A, gene A. Over here, this is gene lowercase a, lowercase a. And down here, let's say this is gene B, capital B. And over here on the pink part, the capital B got switched over to here. While now this capital A has a lowercase b with it, but this lowercase a still has a lowercase b with it, okay? This event right here is crossing over. So that's what happened here. It could happen on all of the 23 chromosomes. It might happen on just the Q arm, it might happen on just the P arm, it might happen on both arms, on both chromosomes. But for this example, we're just going to use one crossing over event and leave this one alone. So to represent that, let's color this part green, and let's put a little bit of pink over here, okay, so that we can show that crossing over occurred. Now, this happens at the very beginning, because remember, prophase, right, is the very first phase of mitosis or meiosis. And so during this first cell division, we're going to have homologous chromosomes. So chromosome number 1 and chromosome number 1 will become separated. The sisters will stay together though. Okay, so let's write over here homologous chromosomes. I'll say homologous chromosome pairs separate. And this means that the cell is going from becoming a 2N cell into a 1N cell, meaning it will only have one copy of every chromosome now after this first division. So let's go ahead and draw this pink one coming over here. So it's a big pink chromosome that has one little green end to it. Okay? And over on this side, we're going to have the green chromosome go this way, because the homologous pairs become separated. So I'm going to draw a dotted line down here, and I'm going to draw a dotted line down here, so that you know that these two are not allowed to go in the same cell, and these two are not allowed to go in the same cell. So this one comes over here. We have a big, okay, got to draw my pink here. And then I'm going to draw that, I'm going to have the pink one go this way, because that'll be fun. We'll do the tiny pink one on this side. And we don't know where they'll line up, so we don't know which one will go into which cell, okay? So I'm just going to draw it like this because it's nice. It makes it a little more colorful. Okay? So we have the big pink chromosome with a small green, and over here we have the big green with a small pink. Now, that's the first cell division. This was a cell that had 46 chromosomes in it still. This was 46 chromosomes. This is still 46 chromosomes even though they're copied, because we count the number of chromosomes by the number of centromeres. Here we have 1, 2, 3, 4 centromeres. Here we have 1, 2, 3, 4 centromeres. So we have 46, we still have 46, even though there's a copy attached. Once we divide the homologous pairs, this is now a 23 chromosome. In other words, these are now 1N, okay, whereas this was a 2N cell. Now, here we're going to do the normal thing that we would do in mitosis. So cell division 2 of meiosis is just like mitosis. These cells are going to line up on the metaphase plate, they're going to be ripped apart during anaphase, and then they're going to do telophase and cytokinesis and end up in different cells. So let's have this long pink half that's just pink end up over here, okay, when this divides. The other half of it that's half green will end up over here. like that. This green one will get ripped in half. Okay, so the green one will get ripped in half, and it will also divide. I'm going to put a little dotted line down the middle here now. And then we're going to do the same thing over here. Let's cut this in half. We're going to have half pink. And then the piece that's all green will go over here. And then the little tiny pink one will go here. And the other half of the little tiny pink one will go over here. Okay? And then we can write here, sister chromatids are separated. Okay, it's 1N, it's still 1N, because we're just ripping apart the sisters. Just like when we did mitosis, we were 2N and stayed 2N, okay? So, let's make sure that we have a definition for what these different cell types are like. Anytime we use the word haploid, that's like half the amount of DNA. So these are all haploid cells. This would be the equivalent of having 4 sperm cells or 4 egg cells that have different variations of DNA in them. So let's go ahead and draw a little sperm cell here. And then let's draw one example, like this one, okay? This would be an example of a haploid cell, 1N. 1N equals 23 chromosomes if it's a human, and it's where we have only one copy of each chromosome. Okay? Whereas a diploid cell would be your normal body cells. A normal body cell would have 2 copies of each, one from your mom and one from your dad. Okay? 2N, 46 chromosomes if it's a human. 2 copies. 1 from each parent. Okay? Now, make sure to answer the last question. You need to use these 3 words in your answer and make sure that you highlight those 3 words. When you are done, please read the differences between how men and women do meiosis. Men and women do meiosis differently because women actually only make 1 giant big egg at the end, whereas men make 4 small sperm, and they can make many more, whereas women can only make 1 a month. I want you to read through that and answer those questions, as well as read through about the egg and fill in this data table. So make sure not to forget to fill in this data table with the information found up in here. Okay? On the next page over, I would like you then to read about how identical twins and fraternal twins happen, and then you're going to answer these questions. Okay? And that is what we need to get done. So, make sure to get all of that done so that you're ready for class on Thursday. Okay, bye-bye.